Hello, I am Himanshu Deka of Faculty of Pharmaceutical Science, Assam Downtown University. In this video, I am going to discuss the third part of my practical, that is study of morphology of bacteria by gram staining. In this part, I will discuss the uh, result and discussion part of this practical. So, in this previous two video of this practical, I have discussed the principle of gram staining, and I have detailedly described the procedure to how to perform a gram staining in a pharmaceutical microbiology lab. So from that we can summarize that gram staining is a process that is developed by Hans Christian Gram. It is a technique that separate the bacteria into two major classes, that is gram positive bacteria and gram negative bacteria. So in this gram staining, we basically use the primary stain, that is crystal violet, and then decolorizing agent, that is ethanol. Then modern we use iodine, and lastly we use the safranin as a counter stain. So after performing the experiment, if we need to, when the slide is ready, we need to study it, we need to observe the slide under a microscope. So after observing, if we get the cell of the bacteria are the color is pink or red, then it will be a gram negative bacteria. If the cell of the bacteria is showing blue or purple color, then it will be a gram positive bacteria. So this is how we have to report this practical. If it is blue or purple, we have to report that the provided microorganism is a gram positive bacteria, or if the color is showing pink or red, then we have to report as it is a gram negative bacteria. So there are some precautionary steps that we have to follow during performing this gram staining method. The most critical step is decolorization by using ethanol, that is ethanol. If the too much, if the crystal violet dye is exposed for too long, to more than 10 seconds, then the primary dye, that is crystal violet, will be wear off and will ultimately get will false result. So we have to be careful while using the decolorizing agent while performing the gram staining. So gram staining, there are various applications of gram staining. It is mainly used to differentiate bacterial species into two large groups, that is gram positive and gram negative. It has some limited use in environmental microbiology. Gram stains are also performed on body fluid or biopsy when infection is suspected. So these are the few applications of gram staining. So that's all for the practical of study of gram staining. Thank you.